All right. So far we talked about basically started with the carbohydrates, to the lipids, and last time we talked about the glycoproteins too. All the macromolecules that are really important and also have two different kinds of moieties, critical moieties like the amino acid or the polypeptide part, as well as lipids or the sugars and things like that. We're going to move on, and basically we'll still talk about the lipids, but more putting the emphasis on their usages in the membranes, as well as proteins that are involved for forming this membrane. Before we get started, actually, there are some critical announcements. After long and deep thoughts, I actually decided to change the grade scale. It's basically just is it me or probably something else? Um, Basically, I mean, I've been talking to the several students and their concerns, especially not being able to come in come in the class because of their family issues or some other like you know, adaptation issues from them. Because it's a huge change, basically, from the last semester. We have online courses to the in-person courses and things like that. Lots of limitations. So I actually lowered the grade scale for everything by 5 points. So instead of you getting 90 to 100 to get A. Now you can actually get as low as 85 to get an A. And there are some clarifications where sometimes your scores are like a 69.999999998 or something like that, right? In which case you were concerned that if that belongs to the so it has to be changed actually to 79.99. So I'll change these. Basically, I just made the change in this morning, so it's kind of looking weird. But for example, this is supposed to be 79.99, right? If you have 79.999, which is higher than this range, so it's going to be round up to the A minus as 80. So that's another kind of plus for you guys. And instead of having all the gradings equally weighted as a 33.3%, I actually made a little change here that the two mid exams will be weighed as 30%. So two 30% is up to 60%. And the final, of course, covering more lectures and chapters will be weighed for the 40%. Any objections? Hopefully not. So, and the question. Yes, yes, of course. So that's one thing. I mean, but this is what the most thing, like the highest bar that I can raise. So don't expect any other changes at all after this. And the final exam is basically on December 14th. So don't be confused. It's on Tuesday instead of regular Wednesdays and Fridays. It's just by the university, so I can't really change this time. And that starts at 8 in the morning. So don't forget, just don't come in 11:40 and then just say that you didn't know about this. So and for 2 hours. So you're gonna have enough time for that. But we'll have the same number of questions as like you do, like 30 questions, and might have some bonus, questions too. Any questions? Yes. So you probably earned some, points up to 10 points for your second midterm, right? So that's gonna be still there. So, but will be, considered for only 30% overall for your total score. And then you're gonna have another bonus of three bonus questions for the upcoming midterm exam too. So that's another plus. So don't get discouraged, even if you have pretty low scores for the first and second exams. Just keep studying hard, and you can still get a pretty good grade here. All right, so we're gonna move on to the actual lecture now. Just a checking time, okay. So today, like I told you, we're gonna talk about more about the constituents of membranes in like more focusing on the protein side, so membrane proteins or some other proteins that are involved in the interacting with the lipids on the membranes and so on. And the most important, thing would be the membrane proteins that are embedded in the lipid bilayer that forms the biomembranes like the plasma membrane or the organelle membranes like ERs, Golgis, and things like that. So these membrane proteins are present in various membrane systems, like I told you, ER, Golgi, subvesicles, and many other things like a nucleus too. And they typically have hydrophobic amino acid side chains oriented outward. And that totally makes sense to make interaction with the nonpolar phase within the membranes. So for the tighter interaction by the hydrophobic interaction, has to have those hydrophobic side chains from the amino acids have to be exposed outside, which is quite different from the soluble protein, right? And because of this hydrophobicity, it's generally insoluble in aqueous solution, and that totally makes sense, as opposed to the cytosolic proteins or the soluble proteins will contain more hydrophilic amino acids on the surface. So that's a huge difference between the membrane bound or the membrane proteins versus the cytosolic proteins. There are different classes of the membrane proteins, and we're going to go over one by one of those classes. They are integral proteins or integral membrane proteins, peripheral proteins or peripheral membrane proteins, and the basically proteins anchored by some other things like sugar or the lipids and things like that. The first thing that we're going to discuss is integral protein, which obviously looking like the stuck right in the middle of the membrane, biomembrane, and they are tightly linked to the membrane using that hydrophobic interaction of the amino acids that are exposed on the surface of the integral membrane, integral proteins. And they can be separated only when there's a special agent that are treated to disrupt the membranes, such as detergent. Has to be pretty potent ones. Otherwise, you can't really easily disrupt that lipid bilayer structure, especially for with the polar reagents or some kind of polar substances. So it has to be something that really agitate the membrane to take out those integral membranes. They're associated again with the strong hydrophobic interactions, and we know why. And because they have the hydrophobic amino acid side chains exposed outside, they tend to aggregate when they're put it in the aqueous solution. And the domain part is especially inside of the membrane is rich in alpha helices and beta sheets. And we talked about the secondary structures. They're basically alpha helices, beta sheets, and the turns and loops and things like that. And as you know, turns and loops are pretty like a flappy, right? It kind of is flexible, as opposed to this integral protein has to be tightly packed to be nicely inserted and stable inside the plasma membrane. So instead of having loops and turns, it mostly have alpha helices and beta sheets. And the removal of this integral protein tends to disrupt the membranes because they're involved in the integrity of the membrane too, by recruiting some other critical proteins, or as they are just working as the regulating the composition of the lipids in the biomembrane. Examples are glycophorin A in human red blood cell erythrocytes. And here's the structure. As you can see, this looking like a long thread or the long chain of the amino acids. But inside the lipid bilayer, it actually forms the nice alpha helical structure, as you can see, as opposed to the exposed regions are more like loose. And another example is a porins in bacterial cells that is pretty much open and the kind of responsible for exchanging the materials inside and outside of the bacterial cells. Second class is the peripheral protein or peripheral membrane protein, which you can see they're sort of relative localization in the plasma membrane or any kinds of biomembrane here. It's kind of stuck on one side or loosely bound actually. It doesn't have to be really embedded halfway of this membrane, it can just softly touch the membrane. It's still called and classified as peripheral protein, and they can be dissociated, easily dissociated from the membranes by simple treatments like treating the high ionic strength of the solution, or even changing the pH, and sometimes temperature too can agitate this interaction between the lipids and the peripheral membrane proteins, because they're not stuck in the membrane, but they're having some like hydrophilic or the H bonding or some kind of other like charged interactions to maintain their interaction or the localization relative to the membrane. And they're generally linked to the other proteins, particularly, you can imagine, the integral proteins, which are stably bound to the membrane. So it uses that as an anchor and have the protein-protein interactions. Again, it's more like the charged interaction or the polar interaction rather than the hydrophobic interactions that are involved for that. Question? That's a good question, actually. Which side? Would that be inside the cell or outside of the cell? They can exist in both regions, basically, but their compositions are different based on what kind of decorations they have, base lipids or the carbohydrates, different kinds, or the functions and things like that. They can localize to different regions or the size of the membrane, biomembrane, which is by lipids. Lipid bilayer, sorry. And they are soluble in aqueous solution because they are less hydrophobic compared to integral proteins. So you can actually extract those proteins pretty easily by just agitating those interactions between the integral protein and the peripheral proteins, and then you can spin it down or use some kind of chromatography to purify those proteins pretty easily with the aqueous solution. And removal of a peripheral proteins leaves the membrane intact, so it won't affect too much in terms of integrity of the membrane. And because it's not really spanning through the biomembrane, but it just is simply attached to that. So it won't affect the overall structure or the stability of the membrane. Example is an cytochrome C, which is involved in electron transport chain, or ETC, mitochondria for the oxidative phosphorylation that we're going to actually talk about during the metabolism. So that's going to be the later part of this class. And cytochrome C, as you can see, is right here. This is a mitochondrial membrane. And the cytochrome C, as you can see, is not spanning through the membrane, but it's just stuck on one side, especially facing the inner membrane space. And there are two more classes, basically. One is the lipid-linked proteins, and the last one is the glycoproteins that we already talked about. The lipid-linked proteins are simply the proteins that have the covalent linkage to the lipids. And these lipids are used as a sort of anchor to interact with the biomembrane in many cases. Not necessarily all the times, but many times it is used as the sort of, some kind of like a stick that sticks into the membrane to hold it right there. And a single protein can contain multiple lipid groups linked together. And all those lipid groups can be involved in the anchoring to the membrane, or have different functions like you're interacting with the other molecules, or they're involved in cell signaling pathways. And they're generally present in the inner leaflets of lipid bilayer. So is that inside or outside of the cell? Inside or outside? That's inner leaflets. Inside, of course, right? So that, going back to the question, where do they have to be localized? And so there's a little preference, based on the what kind of proteins that you're talking about. But lipids generally localized inside nucleus or the membrane. There are lots of examples of the lipid-linked proteins. There are a few of them here. The protein anchored with the myristic acid, which process is called myristylation. It's kind of funny. I use that pretty much every time in my lab, but all the time, but I can't still pronounce it correctly. Myristylation. And the palmitic acid can be also used. And also the process of anchoring the palmitic acid to the protein is called permitylation on the surface of some proteins. Again, this is for localizing proteins to the membrane or interacting with the other proteins or the molecules. Glycoproteins, I don't think I need to explain too much. It's just the proteins with the carbohydrate moieties. And they're generally present on the outer leaflets of a bilayer. And we talked about the functions of glycoproteins already, right? Again, involved in cell-to-cell recognition. So of course, it has to be presented outward, right? To be used for the cell identity recognition, and also cell-signaling pathways and so on. And this is a little complicated version of this, basically. So when you look at the lipid-anchored proteins on the membrane, there are 4 types of anchors that are used for such making the such lipid-anchored proteins. These are 4 of them, which are the amide-linked myristoyl anchors, thioester-linked permetoyl anchors, thioester-linked prenyl anchors, and the glycosylphosphatidylinositol or GPI anchors. And here, what you're seeing are the first and second types of lipid anchorages. So myristic acid and palmitic acids have a pretty similar structure where it has the carboxylate in the end using as a sort of a polar head group. And it has a different length of this hydrocarbon, where the myristic acid is basically C is at 16, as opposed to palmitic acid is a C is at 18, right? So it's only the 2 carbon differences, but it can make the huge difference again. Here's a depiction of the myristylation, where there's a little like a loose protein here, hypothetical protein, that has a linkage to the this myristic acid, which is a little shorter than this one, and this one is the palmitic acid that is linked to the other protein. So again, it's called permitylation. And again, these are the other 2 types of lipid anchorages. So the third one was thioester-linked prenyl anchors. And here's the structure of the prenyl group, where it has the C double bonds, and then 2 methyl groups are attached to the end of these double bonding. And you can see that structure here and here too. So it uses basically this hydrocarbons containing prenyl groups to be anchored to the membrane. And the last class is GPI, or the glycosylphosphatidylinositol anchors. And it, as in name refers, it uses phosphatidylinositol, which is right here. Here's inositol, and then here's a phosphoryl group that are attached to the lipid that has either 2 or 3 tails. And they are attached to the multiple sugars, the simple sugars like mannose, galactose, and so on, to link the proteins to make this lipid-anchored membrane proteins. So those are 4 different types that are all widely used for recruiting the proteins to the membrane. Any questions so far? Okay. And since all these molecules in the end will be just freely bound to the membrane, but it's not basically just restrained by something, right? It's just floating here and there along the membrane surface. So they're gonna behave basically similar to the what other like free molecules do by simple diffusion. So it's only the 2D motions instead of 3D, right? And when you look at all the molecules, so I want to just cover the concept of this simple diffusion first. They are governed by the heat or the energy that is input, and they're gonna exert their movements based on that energy or the heat that they observe in the random manner. And it's called basically, we call it Brownian motion for that. It uses kind of random places like that. There's no specific directions or anything, but there's only one force, which is the differences in concentration, which set up the concentration gradient within the space or the system, and that can actually works as a driving force for the diffusion. So here's an example where you have a water molecule, just a simple water, here, pure water, and then you just drop a dye molecules in there, and then over time it's gonna just spread out in the water solution pretty like homogeneously. And that's the diffusion that drives in this motion and with this consequence. And where these molecules are get together, so they're really highly dense. And then as opposed to the other regions outside of this high density dye molecules will have much lower concentration for that. So there's a concentration gradient to set up, um, and then naturally, and then we'll spread out all the molecules pretty equally. And then that behavior will happen by the Brownian motion. So they're gonna have collisions here and there, but in the end will be like a relatively like spread out pretty equally or uniformly there. And it always works from the high concentration to low concentration, of course. And also the higher the concentration gradient, the faster the net diffusion. And you can imagine that, right? There are more molecules like get together there, which set up the higher local concentration. And then they're gonna collide even more often. So they're gonna push each other a lot faster or more, which will drive this diffusion gets faster. And diffusion evens out the concentration eventually, and that's all about the entropy, right? It's not about the internal energies or anything. I mean, they are indirectly related, but the actual state function that governs in this diffusion, the phenomena, is the entropy that goes higher all the time. And there are different conditions that set up this kind of diffusion, simple diffusion. And they basically create different kinds of forces for inducing such diffusion. And it's basically the flux of those molecules or the solutes, right? So, and also this flow is proportional to the gradient, whether this is concentration or some other forces. It's all about the gradient that's acting between the membranes when you talk about the membrane. And it's across the membrane. It's not about along the membrane, because we just talked about the Brownian motion that is randomly moved along the membrane, right? But when you talk about the membrane fluxes, it's the molecules that is penetrating through, so fluxing through the cell membrane. So one space to the other, like inside the cell to the outside or outside to the inside. And these are the four types of fluxes. So diffusion flux, of course, is set by the differences in concentrations. And that force is called concentration gradient. And also, instead of using the concentration, it can, the voltage changes can set up this flux too, which is called the electrical current. And of course, voltage changes will make it, differences will drive it, so that's a voltage gradient. And also bulk flow will be set up by the differences in pressure. And of course, that's the pressure gradient. And the last one is osmosis flow, which is the same kind of volume changes, but it's by the osmotic pressure change differences, and it also is set up by the gradient. And again, the higher or the more dramatic or the bigger differences in this conditional differences or the gradient, you're gonna get faster diffusion or the more efficient diffusion. And eventually, some molecules that's going through the membrane will be called permeability. And again, this is diffusion, simple diffusion, but its diffusion rate can be different by the molecules that are going through, or the type of membranes that you're using too. And based on those differences in the variables, you can actually have a different permeability too. Permeability is the simple diffusion across membrane, and it's a spontaneous because it's just a diffusion, simple diffusion. So by the Brownian motion, and they do not, the permeability does not require energy. So that means it's not going to consume any energy sources like ATP in case of living system. And the flux is proportional to the concentration gradient because it's essentially the same as a simple diffusion. And there's no saturation is seen. And that means as you have a higher and higher concentration, you're going to have a faster and faster diffusion. There's no saturation point where even if you increase the concentration of your solutes in one side, it will not just hit the maximum and will not increase its efficiency or the speed of diffusion. It's always proportional. And that's because the simple diffusion happens everywhere. The only the barrier is the membrane, that's all. And the flux is, of course, always downhill because it does not use energy at all. So high concentration to lower concentration. And like I told you, depending on what kind of molecules you're using, you can have a different permeability. And hydrophobic substances tend to have a higher permeability, which means that they're moving across the membrane in a faster speed compared to hydrophiles or hydrophilic molecules. And the reason is because in the lipid bilayer, the membrane itself is hydrophobic. So it'll readily interact more easily with the hydrophobes. And those hydrophobes will require lower energies to basically open up the little space between those lipids and go through the membrane. And the first step of this permeabilization or the permeability would be any kinds of solutes that is partitioned from the water molecules because it's just aqueous solution all the time. So it's surrounded by the water molecules. And of course, hydrophobes will be easily partitioned from the water molecules. There's already a repulsion force set up there by the hydrophobic effect. So easily gets to the lipid and will go through that, as opposed to hydrophilic molecules will require energy to tear apart from those hydrogen bonding or polar interactions from the water molecules and then have to get into the lipids. And also it's charged and have to go through the non-polar substance, which is the lipid. So it'll require even more energies, and we'll have much rarer cases where it goes through the membrane. Many biological chemicals are deliberately made hydrophobic to increase the rate of penetration into the cell. And that makes sense, right? Because when you consume drugs, you're not injecting into individual cells. It's just will be permeabilized in the end. And of course, you want to design in a way that it's easily absorbed into the cells. So many kinds of drugs and the pesticides too, DDT is an example, are hydrophobic, so they can easily go through the membrane and gets into the cell to disrupt or help any kinds of reactions there. And osmosis is, as you know, another driving force for the simple diffusion. And it's a sort of a diffusion, but it works in the other way, the opposite way as we imagine about the diffusion of the solutes. It's the diffusion of the solvent, which is basically water molecule. And this water molecule goes across the membrane in the opposite concentration, right? From the low concentration of the solute to the higher concentration of the solute. So it kind of is counterintuitive when you apply the simple diffusion. But think about this in a way that these water molecules are sort of the solutes in this case, because they're the one that they're moving across the membrane. And the high water, low solute, which is in this side, left side, is basically a high concentration of water molecule. And this one moves to the other side, which is a high concentration of solute, but actually low concentration of water. So for the water molecules, it actually still moves to the high water concentration to low water concentration. Does that make sense? Okay. So it's working in the opposite way, but think about the water molecules and their movements. And basically, solutes works as the lowering the Gibbs free energy of water molecules. Because in here, there's so little of a solute, so a lot of water, solute molecules. In other words, there are so much of water molecule, so a lot of water molecule, as opposed to here, just a little amount of water molecule. It sets it's entropically unfavored because there's an order there, right? So the entropy is pretty low there. So naturally, it has to go into the direction where it increases the entropy, right? So Gibbs energy is a setup. Because Gibbs energy, as you recall, is H minus T delta S, right? So entropy is involved there in the opposite way. So entropy has to be increased to get the negative Gibbs energy. And here, to increase the entropy, to have the more homogeneous water molecule distribution, you're going to have a high S, high entropy, which will lower the Gibbs free energy. Okay? And cells basically swell in hypotonic solution where the osmotic pressure, the concentration is basically really low, and it shrinks in hypertonic solutions. So kind of makes sense, right? It depends on the how osmotic pressure is set up in the environment. It will behave differently, and basically there are different strategies in that living organisms use for fighting against this osmotic pressure or the osmotic stress, where we already talked about it, but the cell walls in the plant or the fungal systems or bacterial systems basically have cell walls. So there are rigid structure, so it prevents the cells from swelling, because like with the physical force there. And you remember cellulose is basically involved in the plant cell walls or the, peptidoglycans for the bacterial walls and things like that. You should, which you should know. As opposed to the animal cells use the isotonic conditions where the osmotic pressure is minimized because the osmosis and the concentration inside and outside of the cells are pretty much the same. So it doesn't have to worry too much about the swelling or shrinkage of the cells. And in animal cells, most of the cells pump ions out of the cell because there are a lot of stuff in the cell already, right? So naturally, you're going to have a higher concentration of whatever that is. And colligated effect too, you have more stuff there inside of the cell. So it has to pump out any kinds of unnecessary stuff outside of the cell to basically lower the concentration and lower the osmotic pressure that gets out from the out of the cell. And most important pump for this process, pumping out of the cell, is a sodium-potassium pump that, again, we're going to talk about during this lecture for their mechanisms and their structure too. And we're still talking about the membrane proteins and how they actually are involved in this kind of simple diffusion across the membranes and so on. The membrane proteins that are involved for this simple diffusion are the passive mediated transporters or the passive mediated transport proteins. And this passive mediated transport is also called facilitated transport. And it basically just simply uses that simple diffusion across the membrane for transporting the molecules, critical molecules from inside to outside or outside to inside of the cell. And this, since it uses the simple diffusion, it's the process of a spontaneous and passive transport of molecules or ions. And it goes through the specific proteins called the transporters. And these are basically transmembrane integral proteins, like I said, and they are very specific on substances. That means they're selectively take up the certain substances and they're specialized for. And there are many different kinds of integral proteins that are specialized for certain ions or the molecules. And they're sensitive to inhibitors to react in the protein side chains. And also they do not use ATP or energy source. Again, this is all about spontaneous passive transport by simple diffusion. And this transport rate reaches at maximum. So that means it's saturated when all the protein transporters are being used, right? As opposed to simple diffusion, like in this picture, it's number 3 right here. The molecules go through the lipid without any mechanisms, depending on what kind, how the concentration of your starting point, you're going to have different, diffusion rate or the diffusion speed, and it'll only increase for the function of your concentration, no saturation, as opposed to this channel is obviously used that one whole. So based on its structure of the integral protein, especially inside structure, where it sets the size of the pore, the type of molecules that goes through, and the number of molecules that goes through, it can definitely reach the maximum point where there's just a maximum number of molecules that can go through at a given time. So there could be a saturation for that. And it cannot transport molecules against the concentration gradient because, again, spontaneous passive transport, which is a passive mediated transport. And here's the opposite case where there's an active transport, which uses energy source. In this case, this is an ATP. And in this case, actually, it can go against the concentration gradient. So that can be a peel transport for the active transport. So here are the membrane transport proteins again, and they're typically multi-pass transmembrane proteins. They can still have just a one passage through the membrane, looking like this, and by the alpha helix, like that, like the porins, in the bacterial cells, but also typically have this multiple passages, multi-pass of the membrane to form the one channel there, and that'll actually make the really nice structure here, looking like a barrel, or the channel, where you can create the specific size of the channel or the specific shape of the channel with the different constituents to make it much more specialized for certain transport of the molecules. And the winding of the polypeptide chain is happening there to stabilize the structure, and it allows the hydrophilic solutes to pass through the membrane. So hydrophobes can just go through the membrane by simple diffusion. And of course, you can use channels, but for these channels are more specialized for some molecules that cannot go through pretty easily. So they typically have the hydrophilic inside there. And it also allows the molecules, hydrophilic molecules, go through the membrane without close contact with the hydrophobic lipid bilayer. And there are two main types of membrane transport proteins, which are carrier proteins and channel proteins. Carrier proteins, as their names refer, they carries the molecules. So they bind to the specific substrate or the solute, and then they typically undergo a conformational change to actually leaves the transfer the solutes or their substrate to the other side. As opposed to channel proteins are more like the kind of natural process where they still form the aqueous pores, so they're going to go through the hydrophiles through their channel, but they can happen basically passively too, because it's just an opening there. Doesn't have to really literally carry the molecule there, but they still is pretty specific, so they allow specific solutes. And the channel proteins are much more efficient or much faster in transporting the molecules than carrier proteins because they do not have to go through conformational change or have to bind the solute. So carrier proteins can be either passive or active. And passive meaning that it can just that happens by the simple diffusion or without inputting any energies, or active too. So they can actively select their substrates, bind to them, and then transport it to the other side. As opposed to channel proteins are always passive. And that's why it is more efficient. It's much more faster because it depends on the concentration gradient or any kinds of gradients that sets that determines the speed of this transportation of the molecules or the solutes. So channel proteins, it goes downhills, but carrier proteins, if it is active transport protein, then it will go uphill too. Any questions so far? Okay. So we just talked about the passive and active transport. Again, passive transport is the spontaneous process, and that is dependent on the simple diffusion. And examples of those driving force for passive transportations are gradients in electrochemistry, electrochemical gradient or the pressure gradient, concentration gradients, and so on. Active transport or active transporters actively pump out the solutes or pump in the solutes across the membrane against the gradient. It can be electrochemical gradient or again pressure or concentration and so on. And of course, it requires energy consumption. And it typically happens through the hydrolysis of ATP, where it takes out the phosphoryl group from ATP, which relieves a lot of energy there, potential energy there, and can utilize that energy to move things to the other side. And here's an example of the passive transport. Again, carrier proteins can be either passive or active here, as opposed to channel proteins are always passive, working similar to the simple diffusion. And that depends on the gradient that is set up across the lipid bilayer. For the active transport, of course, it have to use energy, right? But it can be set by the different different mechanisms. Here are the 3 different mechanisms listed here in order for the active transport working. The first one is coupled carriers, meaning that there are at least 2 molecules are involved at the same time for transporting those substrates or the molecules. And the solute is driven uphill, meaning that it goes from low concentration to high concentration. And that's why we're using ATP or energy, and that's why it is an active transport. But it's many times this uphill movement or the transport is compensated by the different solute that is being transported at the same time as in couple downhill though. So it doesn't have to be necessarily all the cases for the coupled carriers, but many times to optimize this energy usage, they have coupled the one that is moving one side that definitely works downhill, and you can actually use that entropy, which can be converted into internal energy, right? To push the another molecule uphill. Another mechanism, the second mechanism, is ATP-driven pump, and of course, it just simply uses ATP hydrolysis for the uphill transportation. And the third one is a light-driven pump, which you can see here. it uses photons as an energy source for the uphill transportation. And an example is the many of the rhodopsins, where you can see it on your eyes and the cells that are responding to the light, and then it triggers the signal transduction to get to your brain or some other parts where you detect there's such amount of light, such wavelength of light is there. So we just talked about these three mechanisms, and we'll delve into these mechanisms again. I know there's quite a lot of information, but they are really important. So of course, understand how this biological system is working and how the membrane is regulated for sustaining life or the signal transduction and things like that, but also important for your exam too. So stay focused. For the coupled carriers, there are two types of coupled carriers, symporters and antiporters. And as their name refers, symporters are the ones that transport the solutes in the same direction. So the first and second solute and third possibly move in the same direction, as opposed to antiporters will deliver their different solutes in opposite directions. So this is an example. Uniport is basically the channel or the carrier that transport just one single molecule. There are coupled transport, the symport, or the symporters will be for the moving two molecules at the same time to the same direction. Antiporter or the antiport process will be the something on the opposite direction across the lipid bilayer. And these coupled carriers utilize this mechanism a lot, like either symport or antiport. And in many cases, they use sodium ion, cation, because the sodium cation is just so abundant everywhere inside the living organism. Oh, question? What type of carrier? I'm not sure if I understand your question. No, it's okay. So what type of? Antiport? Symport? What type of them? Oh, got you. So the question was that the coupled carriers, should they be either symport or antiport, or can they be both? Is that the question, or? I'm sorry. You can ask me later, after this class, because I didn't understand fully what you were asking. But it should be either symport or antiport. There's nothing in between, basically, right? And some molecules, it's pretty rare, but move the three molecules at the same time. So in which case you have to look at all three, the movements of those molecules, and can determine if that's whether sym or anti. And it doesn't have to be, right? It can be both moving inside the cell or outside of the cell. I'm not sure. I'm just throwing a bunch of things and try to answer to your question, but sorry, I didn't understand fully. But anyway, sodium cation is widely used as a second solute. So it's involved with the potassium, magnesium, calcium, things like that. And this sodium concentration is much, much higher in the extracellular space compared to the cytosol. So you're going to have much less sodium inside the cell, right? As opposed to you're going to have a lot in the outside of the cell. And that sets the electrochemical gradient already across the membrane, right? And that is really important. And we're going to talk about that here, that this gradient can be set up directly by the ATP-driven transport, which will be called primary active transport, or primary active transporter can be involved for setting up this gradient, as opposed to, in many cases, actually, this gradient is set up indirectly. So there's an ATP-driven transport that is involved to make this gradient of sodium cation concentration. And basically, this gradient can also drive the coupled carriers, especially for antiport. Make sense? Okay. And that is called secondary active transport because this is not the energy that is input directly to the coupled carriers, but it was set up by something else, which is ATP-driven transport to first build up this gradient, like electrochemical gradient, and then it can be considered as a concentration gradient too when you look at the sodium cation. And then it uses that gradient to drive the coupled carriers movements there, which is coupled to the potassium or some other ions. So depending on where ATP is consumed, you can call it secondary active transport or primary active transport. And this binding of two solutes to the coupled carriers is cooperative. So it happens always, doesn't have to be always at the same time, but one has to bind to induce the other, and it's like cooperation basically between those two transportations. So here's one example of the sodium gradient that is set up. So it's this electrochemical gradient, and also, like I told you, it can be set up, considered as the concentration gradient too when you just focus on these sodium cations. But there's a much higher outside of the cell. So it's already set up by some other ATP-driven transport, right? And after that, this sodium coupled carriers can use this gradient to bind these sodiums there to push it inward inside the cell. But also it binds to the other molecule here. It can be, again, potassium or some other ions that is much rare outside, but still working cooperatively to move all those molecules inside. So this, here's a glucose. This is on glucose actually, it says, which is even if it goes uphill where the inside of the cell has much higher concentration of glucose, it can still deliver that inside, in the opposite way of the simple diffusion. Questions? Yes? Okay. Sure. So the primary, when we focus on this sodium glucose coupled carrier, if that were on primary active transport, then it will not carry this electrochemical gradient. But what it will do is consume ATP, and then will transport both of them to the same direction if they are, if it is a symporter, or the opposite direction if it is an antiport, as opposed to if it is secondary active transport, which is the case, then it itself does not use ATP, but something else used ATP to set up this electrochemical gradient of sodium cation first. And what this coupled carrier uses is it uses this already set up gradient to drive the movement of glucose uphill, the opposite way of the concentration gradient. Does it make sense? Okay. Really good and important example of it's a coupled carrier is a sodium potassium pump. And it's also known as sodium potassium ATPase. As its name refers, it uses ATP. So is it primary or secondary transport? Primary, because it uses ATP. It uses ATP directly. It doesn't need any kinds of other driving force like the gradient here at all. And it's basically this sodium concentration gradient is maintained across the membrane by this pump. And that is, again, a really important process because it sets the electrochemical gradient, which is used for signal transduction. And it is antipolar because it pumps out the sodium cations out of the cell, but at the same time, it ports the potassium ion inside into the cell. And it's pumping both the ions against the electrochemical gradient again, and that's why it's primary, because it really requires energy for that. If it uses one of other gradients, then it would have been just secondary. For the I'm talking about here secondary active transport versus primary active transport. And it pumps out the 3 sodium ions and the intake or the import in the 2 sodium ions. So there are different numbers of ion molecules that are transferred during this process, just one cycle of pumping. Skip checking time. And this pump again is powered by ATP, and that's why it's called ATPase here. And it surprisingly uses almost 1/3 of ATP in animal cell, in an animal cell. So it's a lot of energy to actually consumed by only setting up using this sodium potassium pump to set up the sodium or potassium ion electrochemical gradient there, because it's, of course, is really critical. And for the active nerve cells, it can consume up to 2/3 of all the ATP molecules that are synthesized in the cell, because it sets the sodium potassium gradient there in the nerve cells. If you're familiar with the nerve cell structure, there's axons, and the electrical signal has to basically transferred or transduced through the axon to the next cells, next nerve cells through the synapses and things like that. And this process is really fast. And the reason because is that instead of pushing the protons or the electrons directly into in the cytoplasm inside the cell, it uses in this electrochemical gradients there, quickly releases the ions inside or outside of the cells, set up the gradient and propagate it really fast, quickly there. So that's how axon works, and that's why we're seeing things or moving things pretty quickly, instantaneously. So here's a little mechanism how the sodium potassium pump works. Like I told you, sodium concentration is low inside the cell, but the potassium is high inside the cell compared to those concentrations outside of the cell. And it pumps in 2 potassium cations. At the same time, it pumps out 3 sodium cations there. And ATP is involved, especially for binding the sodium cations. And then it pushes out, and then this phosphate group that was derived from the ATP also helps the binding of the potassium ions. So here's another sort of diagram how this actually works. So here, before we get to this mechanism, this sodium potassium pump can also work in reverse to basically following the electrochemical gradient. And that is the process of generating ATP from ADP. So it's the opposite way of it's working for pumping out the sodium and pumping in the potassium ions. And this pump can be, when it is inhibited, then you're going to have the cells will swell up and burst, and also you're going to have a lot of trouble in signal transduction. So again, how it works is that in the cytoplasm, it has to pump out the sodium ions, right? So it binds the 3 sodium ions in one side, and it actually induces the ATP binding and hydrolysis of that, of this sodium potassium pump, where it induces the conformational change that can easily release these sodium ions outside of the cell. And also, it goes through the conformational change on the other side where it can easily bind the potassium ions here. And by binding the potassium ion, that releases this phosphorus group, and that will also induce the conformational change to going back to its original form here, where it has low affinity to potassium ions, but higher affinity to the sodium ions. So the next cycle begins. So that's keep working basically. And again, ATP is involved. And like I told you in the first bullet point, it can go the opposite way, where you can easily get the phosphorus group there that is freely floating in the cytosol. Then once it have a phosphate, then it can recruit the ADP to make it into ATP. And this process will be spontaneous because this is directly following the electrochemical gradient of sodium and potassium ions. Does it make sense? Okay. And the different type of membrane proteins that are really important rather than pumping or carriers are the channel proteins. And those channel proteins are typically work for ions, lots of small ions. And these works as a sort of a passively. So it allows the passive diffusion of solutes, unlike pumps or the carriers. And some are really large and less specific to their substrates. And it helps for the communications between the cells, used as a gap junction of the two adjacent cells for exchanging their materials inside the cells. And again, examples are the porins in the bacterial cells, and that basically make an opening to the extracellular space. But some are less specific, but most channel proteins are highly specific, and they can also stay in the open or closed conformations, which basically limits those like a free or simple diffusion of their substrates. And they can process up to 10 to the 8th power ions per second, which is a lot, because the most efficient carrier proteins where the ATP is involved, or sometimes not, can have up to 10 to the 5th times faster than just the diffusion. So it's not times actually. It is actually times, sorry. So these channel ions are 10 to the 5th times faster than just the carrier proteins. So it's a lot, much faster and much more efficient in terms of letting through their substrates. And these channels proteins can be used in nerve cells for transducing the electrical signals, which are the action potentials. And we just briefly talked how it works on the axons. And based on what kind of substrates it's going through, it can be named as sodium ion channel, calcium channel, potassium channel, chloride channel, things like that. And there are different stimuli that are involved for inducing these channel proteins to channel through or let through their substrates. And basically, these stimuli changes in the channel conformation to open or closed forms. These are three major stimuli for the channel proteins and for changing their conformation. The first one's changes in voltages. So the one that working based on these voltage changes are called voltage-gated channel. And the next one is ligand binding, where these small molecules, small proteins, or the polypeptides are bound to the either extracellular region or the intracellular region of these channels to make induce the conformational change for open or closed conformation. So they are called as ligand-gated channel. And the last one is mechanical stress that like mechanically force the push away these channels, make this opening wider or smaller. Um, and they are called mechanically gated or stress gated channel. Question. So for the question was that what is the mechanical stress actually? What are the mechanical forces that can be applied there? Of course, we can't do that like with your fingers and things like that, right? Those are the proteins in many cases that are just sitting there, and based on their conformational changes or the number of proteins that are sitting there, they can actually make it open. And they have some polymer structures that can be involved there too. So those are the major forces there, and also it can be literally the mechanical force where the some cell detects the pressure or some kind of wall or mechanical force. Then you want to respond to that, right? So in which case it really literally detects the mechanical force there, and then induce this conformational change. All right, so these ion channels have lots of different types. More than 100 types are discovered based on their substrates or the molecules that they're going through, let through. And but the common ion channels are mostly responsible for the potassium transport. And some are in the open conformation continuously. An example is potassium channel, and it's specifically called the potassium leak channel because it is basically leaking. It's always open. And it's important for maintaining the voltage potential or the electrochemical potential across the cell membrane. And the concentration of these potassium ions that we've seen, there are more inside the cell and there are less outside of the cell, is kept in equilibrium by this K potassium leak channels. So because you can't, always just build up the higher and higher potential there, you have to just maintain it at a certain level to make it most effective to respond to the any like neuronal response. So the potassium leak channels does that, keeping the equilibrium. And sodium potassium pump maintains osmotic pressure. We talked about this, but specifically when we look at the number of molecules that are transported, the sodium molecules are pumped out basically to the cell, but there are 3 molecules there as opposed to only 2 molecules of potassium ions were transported into the cell, right? So when you just count the number of cells, you're losing 3 cations, but gaining 2 cations. So that's going to be -1, actually. You're losing 1 positive charge. And that is pretty critical because it balances the anion cation concentrations within the cell. Typically, the most organic molecules have the positive charge, cations. And you, as you see, I mean, you have a lot of like cations involved for the proteins that are bound to proteins. We talked about the hemoglobin where the magnesium, the cations are involved there. So there are lots of small cations there. And there are too many, right? So it actually makes it unbalanced. So it has to be pushed out. And obviously this sodium potassium pump can lose this positive charge there. So you can actually keep it at a lower, relatively lower positive charge, overall charge inside the cell. Here's a potassium channel again. And there's a really interesting fact in here where what it actually determines the substrate of this potassium channel. This potassium leak channel that we just talked about is really specific. So it only allows the potassium ion to go through that channel. But there are a bunch of other cations that have similar properties there, especially when you take into account sodium cation. They're both small spherical positively charged ions. And when you look at the radius of those two ions, they're quite similar, as you can see here. Not much of a difference there. So it is really weird. And just looking at the radius, the sodium ion, the radius of the sodium ion is even smaller. So that means potassium channel simply won't work by selectively, select for the size of the ions that it goes through, because potassium is bigger, but the sodium's not, so ions not working there. So there got to be another specific mechanism that is involved for selecting this potassium ion. And that is not about and it's also like, it is a channel, especially leak channel. So this, passage, the letting through the potassium ions are really fast, right? So it's not about specifically selecting and binding the potassium ions. So it's not about, and also it does not use ATP, any energy source there. So it's not about using energies to identify the potassium ions. So it's got to be something else. So it has been a conundrum for a long time until we have determined the 3D structure of a bacterial potassium ion using the X-ray crystallography. And here it actually shows the little bit of mechanism and the structure too. But by this X-ray crystallography, we could determine the 3D structure of that channel, and found out lots of things about that to explain why it is so selective for the potassium ion. And that is, this potassium channel, leak channel, has 4 protein subunits, and what you're seeing in this picture are the only 2 subunits, and rest 2 are going to be on top of this, like vein, like the structure here, and the behind of it too. And when you look at the charges of the channel, there are many negatively charged amino acids that are on the cytosolic end of the channel, which is on the bottom here. So they're negatively charged, so they're going to naturally attract the positively charged ions, the cations there. And inside of this channel, there are lots of O's here, oxygens. And these are carbonyl oxygens from the amino acids basically here. And they line the interior of a pore with the really precise like configuration there. So there's a precise separation there. This is a top view of that carbonyl oxygens in the core of the channel. And they maintain this specific like size or the relative location there. And that actually makes this channel so selective for potassium ion. When these ions are floating in the cell, because it's an aqueous solution, so they're going to be surrounded by water molecules and naturally make this kind of hydrated form of the ions. And this will flow, like randomly inside the cell. And once it goes closer to this potassium channel, then it'll be attracted by these negative charges, right? And once it gets closer to that, gets into the pore, and then this carbonyl oxygens, which are sort of negative, are naturally attract these cations through the channel. But here, still, these cations are surrounded by water molecules, and they have specific conformations like that. And this carbonyl oxygen lining is exactly mimicking these water molecule interactions. So it'll spend minimal energies and pretty favorable to changing from this conformation, the hydrated form, to the just ion moving into this channel, inside the channel. And that will just simply go through up to that, and that will release on the top of this potassium channel. As opposed to sodium ions, it also forms this sort of a tetrahydrated structure here. So it'll be attracted by these negative charges on the bottom of the potassium channel and then gets into that. But once it gets to the this inside where the carbonyl oxygen lining is there, then instead of having all four carbonyl groups that are interacting with the sodium precisely, since it is a smaller, it'll interact only with the two of them, right? So two interactions versus four molecular interactions with water. So this one be more favorable. So instead of going through this, the cavity here or the channel through here, the sodium ion will go back and will try to be connect with the water molecules. So that's the core mechanism there. Does that make sense to everybody? So even if sodium is smaller, still it's not optimized. It's not the most favorable condition where it binds to these carbonyl oxygen groups. All right. And the carbonyl oxygen groups that I talked about inside the core of this channel is called selectivity filter or ion filter that actually awarded the Nobel Prize in Chemistry in 2003. And it was based on the discovery made in 1998. So it's only 5 years before. So that was a really, really important finding then. So even if after 5 years, everybody realized that this is just a so critical finding. And that is basically purifying, or not the purifying, but determining the structure using the X-ray crystallography of this ion filter or the selectivity filter using the bacterial potassium channel. And that explains everything about that selectivity of the potassium ion over sodium ion. Okay? And especially for these channels and carrier proteins, like all the membrane proteins, they cannot simply just translated and stuck in the membrane. It's just energetically so unfavorable because you have to just push the bulky protein into the membrane, and there are some hydrophilic regions there too, right? So instead of generating some soluble proteins first in the cytoplasm and push it into the membrane, like many of the other proteins, it actually generates the transmembrane protein even from the beginning in the ER. So how it does is that it gets to this mRNA first, and that is transported to the rough ER for its translation, right? Normally, it would have been just translated and released to the cytoplasm, but instead of doing that, it contains the signal peptide region, which is translated first, and then that will be detected by this SRP protein, which contains a GDP, and that becomes the GTP, and it will actually stop the synthesis until it gets to this translocon on the rough ER. Once it meets the SRP meets this translocon with the SRP receptor, then the translation resumes and cleaves this signal peptide, which is not necessary anymore. But the translation is keep going, and as you know, there's a co-translational folding. So there are a lot of foldings happens, and the sugars attached there, carbohydrates are attached there, as well as some other things can be basically modified. Ended up having this transmembrane protein. And this transmembrane protein on the ER is going to be released in the form of a vesicle from ER to Golgi to the cis-Golgi network, or CGN, and then will be further processed through this Golgi to the trans-Golgi network, or TGN, and eventually will be released in the form of a vesicle again to the plasma membrane to get into that. So it's the fuse of vesicles that consists of the lipid bilayer already, right? So lipid bilayer and lipid bilayer fusion will be much easier than inserting the protein into the membrane. So that's how it works. And also it can be secreted outside or degraded by using these lysosomes too. All right, so that's it. And we're gonna have a review session, next Wednesday, which is also really important. So, but my, lecture notes and the presentations are the most important things. So go back and review those for your, preparation for your exams. And good luck, and we'll see you next Wednesday.